Hi, everyone, and welcome to the talk today that I'm doing on personalized nutrition. And specifically, we're going to be talking about personalized nutrition as a part of a personalized medicine framework. So I'm Geere, and I'm going to start with my disclosures. So first with non-financial disclosures, because I just have so much gratitude for these places that I've worked in the last few years that have led to this talk. The first being Jumpstart MD, which is a private medical practice here in the Bay Area that specializes in personalized nutrition. Genova Diagnostics, that is a laboratory that I'm going to get into a little bit later in the talk that does testing on personalized nutrition. And 100 Plus Labs, that is a startup that is looking at longevity medicine and coaching and the utility of personalized nutrition. Financially, I have started my own companies here that are listed. The first being a tech startup. It's basically a laboratory testing platform that delivers mobile application for laboratory testing. The second being a practice where I deliver personalized medicine and nutrition. And the third being a program that is a nutrition program designed based on some of the topics we're going to go through, specifically around circadian rhythm and how that applies to nutrition. All right. So one of the most important things to remember is that no two people are the same. And diets today are designed to essentially fit sort of a one size fits all, right? And this is really important to keep in mind. So when you're working with a dietitian, at least today, what we're finding is that people are giving generalized advice. And the recommendations are not really built based on science and what is currently taking place in the literature. And so it's very important to start to address this problem because we have the obesity epidemic and chronic disease essentially making up 50% or 40% of the world's population when it comes to disease, right? It's not just a US-based problem anymore, it is worldwide. So we're addressing this problem with essentially the wrong tools. And what's so nice about laboratory medicine is that we get to reevaluate these tools and see if we can start to bring some of these out into clinical practice. So today I'm going to talk about some of the studies and the research, giving you more background. We're going to start with looking at continuous glucose monitoring and some of the data that came out. Specifically, I want to talk about one study that was published in Cell. We're going to focus on some of the instrumental studies in this field. The other one is the gut microbiome. We can't talk about personalized nutrition and precision nutrition without talking about gut microbiome. And then we're going to get into some of the different debates out there about saturated fats. And I just want to present to you some of the leaders in this thought process. We'll talk about plant-based diets, and then we'll get into some of the genetics, the circadian rhythm, and I'm going to end with a case scenario of how you can actually use precision nutrition in preventative health and wellness. So we have limited time, but I'm going to quickly move through some studies just to give you an overview. We'll start first with this paper that was instrumental, published by Aaron Sagel and his group from Israel. And they went on to do more research even beyond this, but this was published in Cell in 2015. This study is essentially looking at 1 week of a dietary intervention, and it was very methodical. They looked at overall 800 people and in a cohort, and they measured about 50,000 meals, but really feeding people specific food for 1 week. And they highlight this in figure A on the right over here. And you can certainly go look up this paper afterwards if you'd like more details. But what they're doing is they're feeding people in duplicate. For example, day 1 and 2, they gave them bread. Day 3 and 4, they give them bread and butter. 5 and 6, glucose, and 7, fructose. And then they measured through continuous glucose monitoring what their essentially postprandial glucose response was. And essentially, bottom line in this study is they found that no two people had the same response to the exact same diet. right? And yet we are giving people dietary advice that is essentially universal. There are choices. However, we're telling two people to eat the same thing. We're not looking at the personalization. And so this group, Aaron Thigl, went on to actually develop an algorithm because he studied people's blood tests, their gut microbiome, did questionnaires on them, anthropometrics, and then essentially designed an algorithm to personalize the information that they were getting or the guidance that they were getting, and then went on to do more studies to show the outcomes of that, then validating their algorithm, essentially turning it into a commercial product. But that study was essentially the foundation of it. We're going to switch gears a little bit. And now that we talked about glucose monitoring, move into mouse studies, specifically looking at the gut microbiome and some of the influential folks here. So in nutrition and gut microbiome, you can't talk about that unless you talk about Jeff Gordon's work, because Jeff Gordon really is one of the fathers of studying the microbiome. And we actually have Peter Turnbout here at UCSF that is continuing his work, was originally a postdoc in his lab. So this is one of the studies where this group came out in 2005 and did some very important work where they looked at obese mice and studied their microbiome and found that there were actually differences in terms of their microbiota when they were all fed the exact same diet. And they studied what these microbiome changes were, and they found at the Bacteroidetes and the Firmicutes percentages, essentially their ratios had changed when the same diet was fed to obese versus lean mice, right? And so this really started sort of a lot of research that then came out of some subsequent studies in this lab. They then studied humans and looked at twin studies, and also found here that using 16S RNA at the time, which then they went on to do more whole genome sequencing, they found significant diversity between twins as well, right? So it wasn't just a genetic issue. There was actually diversity in the microbiome. And so this is some of the findings from that study, that basically showing that although human microbiome is shared amongst family members, that each person's gut microbial community can vary in the specific bacterial lineages that are present. And there's a wide array of shared microbial genes amongst sampled individuals. So essentially, you can look at this at a genetic level and say that there is a core microbiome that is present, but above that, there is a lot of variability. They also then showed that obesity was associated with this phylum level change in the microbiota. And I'll show you later that many studies validated that this phylum level microbiota is altered dramatically by your diet. And it's not just the macronutrient composition of your diet. There are many things that go into it. And so obesity is one disease condition, one condition we can look at. We now know that this actually affects multiple disease conditions, including infectious diseases and immune function. So the same group went on to do even more interesting studies where they took the twin feces and actually transplanted it back into the mice. And so this is a study where they looked at 4 twin pairs, and these twins actually had discordant obesity patterns. So one was obese, one was not. And then they took recipient mice and essentially transplanted the feces back into them, studied them over about 35 days using RNA-seq, and found that the mice actually gained weight when they got the transplanted obese genome microbiome, and essentially increased body fat mass, and the lean one did not. Right, the lean one did not have that outcome. So they then went on to study the actual microbiome composition. It was a similar finding where the Bacteroidetes and the Firmicutes ratios had changed. So the outcome of this is that there's a transmissible, rapid, and modifiable effect that diet has on the microbiota, right, within 35 days in these mice. So we're going to switch gears a little bit. Now we've talked about the microbiome and the role that that's played, how we got to understanding the inter-individual variability from the data in Israel. And now we're going to talk about some of the more controversial topics that are out there. One particular one is about fat. I had the fortune of actually doing some work in ketogenic diets and working with people on fats, and saw some dramatic changes in their biomarkers and health outcomes, and actually got introduced to some of these researchers, including Jeff Volek and Ron Krauss, who are also fathers of this work, but not in the microbiome area, now specifically proponents of saturated fats as a good thing for dietary modification. So this is a paper that they came out with, a recommendation in the Journal of American College of Cardiology, where is most of where we find the discrepancy, where people are told not to eat saturated fat. And so essentially the current guidelines, the previous guidelines were that US dietary goals were essentially less than 10% of calories from fat. However, what we're finding is that when you really study fat, there's a different biological effect based on the type of fat you're looking at. So you can't just put it all in one category. And that's kind of the underlying problem with most nutrition studies. And several foods that are relatively rich in saturated fats, like whole fat dairy, dark chocolate, unprocessed meats, are not actually associated with increased cardiovascular or diabetes risk when you actually look at the studies and do these trials. So there's no robust evidence that the current population-wide arbitrary upper limits on saturated fat consumption will actually prevent cardiovascular disease or reduce mortality. And so this is really important, because as a result of these recommendations, we have started to, we have essentially the last 20 years lowered something that in your diet will have increased your ability to feel satiated, right? Fat actually increases satiety, and therefore people are having low fat, high carbohydrate, high sugar meals and gaining weight, getting more diabetes, getting more cardiovascular disease. Well, what about plant-based diets? So plant-based diets are all often quoted in literature to be healthful, and they're certainly not that easy for a lot of people to follow. When it comes to the health benefits, there have been about 200 studies or so that have been recorded and analyzed in this paper. And these are just the general overall outcomes that we know of so far in terms of some of the positive changes. And these are on average about 24 months where people have been on a plant-based diet. So overall, lower fasting glucose, increased insulin sensitivity, lower LDL, increased short chain fatty acids, all beneficial outcomes, lower BMI, lower chronic disease like obesity, cardiovascular disease, lower risk of certain types of cancer, which I'm going to talk about in a second. And then also changes in the microbiome. So increased Prevotella and decreased Bacteroidetes affected essentially from the plants that people are eating. And then interestingly, some cytokine changes like reduced CRP, reduced IL-6. And this one was specifically looking at the gut-brain interaction and looked at the hormones as part of their analysis and their systematic analysis, meta-analysis. They showed that there was a change in emotional health, in motivation, cognitive features, and then also brain activity. Now, what about genes, right? We've talked about the microbiome, we've talked about different macronutrients so far. What about genetics? How do genes play a role here? And when we're talking about genes and diet, it's actually taken a whole different perspective now, because a lot of these studies are association-based studies. They're not, they're looking at big data, essentially looking at correlations and looking at different genes. And so we are really at the beginning stages of understanding and doing this in terms of clinical trials and how to use this data. But the associations are still very interesting. And there are companies that I'm going to show that have actually gone on to take this to the next level of actually implementation. So nutrigenomics, in this case, we're going to look at its effects on breast cancer as just one perspective. This is a a summary of different studies. And if you look at I've quoted everything, all my literature. So if you look at the links, you can look at the original data as well. So always be, skeptical about everything you read. But here is some summary that's there provided in this particular paper. So BRCA1 and its association with different food, essentially, products are listed here. So I'm going to focus on just a few things that are highlighted. So, for example, cobalamin and folate intake in the presence of the BRCA1 gene, essentially, we're looking at proper methylation status, right? And so you have lower risk when you have those two together. Whereas something like antimony is actually found in a lot of food packaging. So just like arsenic is, so is antimony. And unfortunately, it is still out there and being used. And in the presence of this BRCA1 mutation, the presence of antimony, you actually have an increased risk of breast cancer. Soy is actually very much a controversial topic for a lot of people. They're still under the impression that soy intake causes breast cancer. However, when you look at the nutrigenomic data, in the presence of BRCA2, soy intake, as well as one of the soy products, genistein, have actually reduced the risk of breast cancer. Other things like alcohol is often a question that people have. So what about alcohol? In the presence of the CYP19A1 SNP, if you have the homozygous variant allele, in the presence of drinking alcohol and that allele, you do have an increased risk of breast cancer. So this is, you can imagine, this is how you start to apply some of this data when you are working with patients. EZH2 is actually a histone and a product modifier, essentially enzyme. So this SNP is going to affect post-translational modifications and essentially epigenetic changes. So you've got the presence of omega-3 and polyunsaturated fatty acids with this particular gene is going to lower the risk. MTHFR is a very well-known gene in the nutrigenomic world. This one, when you're looking at this particular mutation that's listed here and folate uptake, you actually have a higher risk of breast cancer. Whereas when you look at certain vitamins that might be actually missing, right? It's not the folate that's causing the breast cancer, it's the mutation, right? In the presence of, even if you give people folate supplementation, it's often not enough because of this mutation. So low amounts of B6 and low folate actually have patients with those levels actually have an increased risk. So you can imagine working with a patient with breast cancer, you'd want to make sure you're addressing vitamin B6 and folate, and you're going to want to make sure you're looking at this genetic SNP. It's not yet being done in practice in conventional medicine. The NAT1 and 2 are essentially a genotype that's associated with red meat consumption. So, there's again controversy around red meat, whether or not that's good for you. Well, in the presence of this SNP might not be such a good idea. So that's just a general overview of how you would potentially use some of these SNPs in a condition. You can imagine, as we understand the associations better and start to subclassify our different disease conditions, we'll have more information and we can do better studies. The other thing that's really interesting in precision nutrition these days is the effects of time. And daytime, when in the day do you eat? And this also is a product of fasting studies, right? So fasting studies, as well as when to eat, and this field of nutrition that's called time-restricted feeding. You're going to hear a lot about this if you haven't already, because the data is very interesting. And Dr. Sachin Panda from the Salk Institute is doing an amazing job with looking at animal studies and specifically mouse studies and looking at time-restricted feeding and publishing some wonderful data. But this really comes down to the circadian rhythm and understanding the hormones in the body and when during the day these hormones peak. And so just a reminder for those of us that might have forgotten is that every organ in the body essentially has a clock, a circadian clock, and we are programmed to eat during wakeful hours. So just the idea of changing your biological clock and working at night, staying up all night and sleeping during the day, you can imagine that it's going to have metabolic effects. And this is what is seen in the literature as well. In Panda's studies, he looked at lab mice in different groups, and where essentially one was allowed to eat whatever they wanted whenever they wanted. The other one had a very restricted 8 to 10 hours, sometimes 12-hour window. He tested different window times and essentially showed that even though they're having the same calories and were given the exact same food, just by restricting the time, there were significant differences. So these are some of the variables he measured, and the significance on the right, the p-values, and you're looking at week 8 changes. So in 2 months, there was a reduced amount of hemoglobin A1C percentage. There was a reduced fasting glucose, and obviously changes in BMI, total fat mass, and visceral mass. So this is looking at the animal studies. I want to move on to how you would apply something like this in a clinical scenario, because that's always the question. It's like, okay, you know, we have this great data. Well, what's actually happening in practice? So again, I'm going to talk about this at the end. We haven't quite gotten to the point where conventional medicine is adopting this. However, it is being done in alternative medicine, in integrative health practices, and specialty private practices. So I'm going to give you an overview of how some of the data is being used. So this is a case of a 31-year-old female patient. She was an avid athlete, cycling 2 to 3 hours a day, long distance races, you know, cycling from San Francisco to Hawaii, those kinds of races. She weighed 230 pounds, and she could not lose this weight. She was this weight since her 20s. Her mood was incredibly labile, crying all the time, and had a history of eating disorder. So when you if she were to go to see a primary care doctor, they probably would only do a very limited workup and tell her they didn't, you know, that she needs to see a psychiatrist essentially. However, if you look at her data a little bit more closely, you might notice a few interesting changes. So when you apply some of the nutrigenomic data, as well as the gut microbiome testing that I'm going to talk about later, you can actually see some of the early shifts that are happening in her body. So her total cholesterol was 198, still below 200, but not ideal for her age and her athletic output. Her CRP was 4.9. So certainly elevated for her. And then when you look closer at her thyroid and check for thyroid antibodies, she actually had elevated thyroid antibodies present. She also had changes in her gut microbiome. Again, I'm going to show you some of that, the companies that do these studies, but she had low beneficial bacteria and essentially poor digestion. So their actual metabolites that were measured from the feces showed these outcomes and changes in her gut microbiome, as well as then the effects. As you can see, she's developing an autoimmune condition and has an autoimmune condition essentially in her thyroid. So she's got this gut autoimmune change that's happening, as well as the elevated CRP. So her plan was, you know, over the course of 6 months to work with a nutritionist and health coach to make some of these changes, as well as a few supplements and changes in her lifestyle. So essentially she modified her diet pretty significantly, got rid of gluten, dairy, and eggs. There's some data that I did not show that was basically based on some food sensitivity testing that was done, as well as just changed her overall exposure to xenobiotics, had more pre and probiotic foods in her diet. She went ahead and monitored her body composition, actually decreased her exercise, increased her sleep, decreased her stress, was put on high dose probiotics and some other supplements for her gut, like L-glutamine, curcumin, ginger, and fiber. She was dosed with high dose vitamin D. A couple of data points that were missing in the last slide. She had low vitamin D levels and actually showed up to have a vitamin D receptor polymorphism. And she also had MTHFR gene, mutation, a homozygous mutation. And so she was put on these methylation precursors that are different from actually just giving her folate. There are some of the precursors that will be better metabolized when the enzyme is defective, when the MTHFR enzyme is defective, when you have those mutations. So as an outcome of 6 months of a diet and lifestyle program, her weight dropped from 220 to 177. Her blood pressure dropped to 98/57, more appropriate for the type of exercise and activity she's had in her body, or the effect that's had on her body. Thyroid antibodies completely normalized. And most importantly, she felt renewed, calm, lost her anxiety completely. She was completely calm and felt for the first time in her life, she felt normal again, and that she could have a whole and complete life, which was huge for a person like her. Now, I referred earlier to some of the studies, I'm sorry, some of the test companies that are out there, and I will, you'll have access to this later. Certainly you can look them up as well. The first one I just wanted to talk about is the Nutrigenomics SNPs. So this is a company that's out of Toronto, University of Toronto, actually, where the researcher went on to create a product where he took a lot of his research on SNPs and put them into a lab test that you can order out of U of T. And it's called Nutrigenomics, and it'll give you nutrition SNPs, all pretty well validated in the literature, and as well as some exercise and performance SNPs. The GI effect stool analysis is from the company that I mentioned in the beginning that I worked for, called Genova Diagnostics. I have no financial relationship with them, but they do have a pretty good lab test that is clinically run by many practitioners across the world, that's looking at different metabolites that are being shown, at least in literature, to have a positive benefit when you modify them in people's diets on the gut microbiome, as well as just general gut health. The same company also produced a metabolomics product more recently that I don't have any experience with, but does some nutritional testing. And the one at the top is the DNA company, also looking at SNPs, and they have some SNPs around the circadian rhythm, and specifically looking at that. So in the interest of time, I'm going to not get into the actual sample reports, but if people have questions afterwards and want to stay over, we can actually look at some of those sample reports together, which are linked over there. Just future considerations to think about is, well, how does all this information, it's great research, good literature, and it's been going on, like I showed some excellent research since 2005. How do we bring this into practice and start to, bring in pretty benign changes in diet and lifestyle that can have some pretty profound effects on disease? How do we start to integrate this? So currently, we're often integrating nutrition and nutrigenomics, like I mentioned, as well as now some microbiome data in private practices and integrative health and wellness practices. This is happening across the country as well as worldwide, including Asia. But there are still very much separate approaches between what's being done in private practice and what is covered by insurance-based conventional primary and especially medical practices. Whereas the literature is showing that there's definitely reason to start to introduce this also in chronic disease, that is where patients are being seen in these conventional practices. There's also market pressure where you have a growing market of health coaches. We have more and more people that are working with health coaches and functional medicine practitioners. So the demand is high from the patient standpoint, but the system is not yet built to support it. So really, how to bridge that gap is we need more clinical utility studies. I had the fortune of working for the Institute of Functional Medicine, where we trained doctors to use some of this information in practice. And what is missing is the evidence base around clinical utility. It's often got good analytic data for a lot of these tests and metabolites, but when it comes to practice protocols and clinical utility and outcome studies, that is missing. And that's really what's needed in order for adoption to occur. We also need more drug supplement interaction studies, because What we don't fully understand is what doses can you give people when you supplement, and what doses are effective, and how do those doses interact with the different drugs that people are taking, because that's often where the errors start to occur. There are a few practices that are doing this. One of them is Parsley Health, another that isn't quite alternative functional medicine yet, but is certainly looking at more interesting data as go forward. These are both relatively young practices, but are certainly making headway. Parsley is doing personalized nutrition as part of their practice, and they are insurance-based, and they've reduced the cost of care. So they're really doing an excellent job with it, but they're really the only ones right now that are doing this type of medicine. And so the future really includes the integration of precision nutrition into things like chronic disease specialty centers, like cancer centers or autoimmune condition centers that are focused on autoimmune conditions, or gut health, like GI effects could be very much utilized in GI specific centers, as well as fertility, where we have a growing amount of people interested in fertility, and definitely nutrigenomics plays a role in fertility, yet it's not being incorporated into practices. So the future really lies in integration of these basic modalities back into conventional medicine, and also in longevity medicine, and essentially applying this back to how do we help people live longer, live healthier, and prevent disease? And this is, again, another new field called longevity medicine, which is growing, and there's more people moving into this field as we live longer, as we get, good treatments for cancer and for other conditions, help people live longer. How do we help them live a healthier, longer, happier life? So with that, I'm going to stop. We're 2 minutes over. And if anybody has any questions, or we can look at some of those sample reports as well.
Thanks, Dr. Geere.